So good evening, everyone. I'm Karthika. I work under Dr. Harsha Bajaj. I'm from CSAR NIIST Trivandrum. So today I'll be talking to you about membranes and biomolecular condensates, how they interact with one another and how do they influence each other. So I'll give a brief introduction about biomolecular condensates. So as we all know, the cellular interior is highly compartmentalized. And in addition to membrane-bound compartments, there exist several membraneless phase-separated compartments inside the cells. So a number of proteins are nowadays being discovered to undergo liquid-liquid phase separation, and they're involved in a number of functions inside the cells. So for example, you have stress granules, paraspeckles, nucleolus, to name a few. In addition to that, there are a number of several membrane-bound proteins which undergo LLPS, and they are also known to interact with membranes. So the major property of these condensates are that they are dynamic in nature and they lack a membrane. They are known to sequester specific biomolecules inside them, hence giving them a spatial and temporal regulation over different cellular functions. Now, the key challenges in understanding LLPS compartments inside cells is, one, like I said, is their dynamic nature. They have very low life, or like very less half-life. They form and dissolve back very fast. Another one is their small size. They're also referred to as punctas because of their minute size and their complex compositions. They lack a boundary, and also the crowded cellular environment makes it challenging to study them inside cells. So what people basically do is they isolate LLPS forming proteins, or they make liquid-liquid phase separated droplets in vitro systems and study their properties in isolation. So in our work, we use a synthetic cell platform or an artificial cell-like system inside which we assemble condensates and then study their properties. So the major questions that we try to answer in this work is, do membranes have a role in controlling the dynamics of condensates? And do condensates in turn have a role in reshaping or remodeling membranes? Does electrostatic play a role in governing the kind of interactions membranes and condensates undergo? So let me explain how we do this. So for the condensate systems, we chose a peptide and nucleotide-based condensates, which is composed of ATP, which is the negative counterpart, and polyl-lysine, which is a positively charged polypeptide. Under appropriate buffer conditions, they form these beautiful condensates. For the membrane mimicking system, we use giant unilamellar vesicles, which are basically phospholipid vesicles that are micrometer sized. You can also imagine them to be like hollow cells. So basically, we assemble these condensates inside these vesicles so that we can study them in a cell-like microenvironment. How we do this? So basically, we encapsulate one of the component, that is polyl-lysine, inside our vesicles, and we add the other component, ATP, from outside the vesicle. This gives us a spatio-temporal control over the assembly of condensates inside our vesicles. So we engineer our vesicles in such a manner that they are selectively permeable to small molecules. So because of which ATP is able to transport to the interior of the vesicle. Once it is inside, it can combine with PLL to form these condensates. Now, how do we make them interact with one another? So we regulate the interaction strength of condensates and membranes by simple electrostatic forces. So we can actually manipulate the charge of membranes and condensates by manipulating the components that we use. So we add, what we do here is that we add a positively charged lipid called as DOTAP. And when you increase the concentration of DOTAP, you make your vesicles more positively charged. Similarly, you have charged counterparts in your condensate system, that is ATP and polyl-lysine. And if you increase the concentration of any of the components, you will get either negatively charged or less charged condensates. Now, if you try to see their interaction in different permutations and combinations, you will get situations where they have high interaction with each other and conditions where they have low interaction with each other. So in this recent work, what we have looked into is, how does the interaction strength between condensates and membranes result in the kind of structures that we see, or how do membranes regulate condensates based on the interaction strength? So what we see over here is two entirely different morphologies. One condition where you have high affinity or interaction between condensates and membranes, which means that one of the components at the vesicle is highly positively charged and the condensates are negatively charged. In these situations, you see the size of condensates being regulated by the membranes. On the other hand, where you have least interaction strength, there is no change in the dynamics of condensates. They are able to grow. So this is one of the inherent properties of condensate, that they will fuse over time and they will grow in size. So here what happens when it has least membrane affinity, it can grow in size. There is no dynamic regulation, and then it generates a force on the membrane. We call it as membrane budding. So I will explain each cases, why and how it happens. So in the first case that I mentioned, that is budding, what happens is that, sorry, just a minute. Yeah, so as you can see in the live process, we have polyal lysine encapsulated inside our vesicles, and once ATP is added, condensation has started. So in this case, yeah, as you can see in the live video, the condensate is able to grow in size, and once it grows in size, it generates a force on the membrane to cause the membrane to form a bud-like structure. So here, there is no restriction for the condensate. Therefore, it is growing in size. So this happens, like I said, in conditions where there is least interaction or least affinity between the condensate and membrane happening. So we analyzed the diameter size of buds, and what we saw was that it is always larger than the other morphology for the condensate that is causing the other morphology. Wrinkling, on the other hand, the other morphology, here what happens is that you have your polyal lysine encapsulated inside your vesicle, and then when you add ATP, the nucleation of condensation occurs at membranes itself. Once it starts, you don't see them moving anywhere else. They get stuck on the membrane. So what happens is that, so you can look at the video. So what happens is that once they start nucleating on the membrane, they stay there. They're not able to fuse with each other, so their sizes get regulated. And wherever they are interacting with the membrane, you see a local fluorescence intensity getting increased. So we were very intrigued by this kind of a reaction. So if you see in this video, we see puncta-sized or very small, minute-sized condensates that are getting sequestered on the membrane. Even though they are in very close proximity, you don't see them fusing. So we were very intrigued why this is happening, why the sizes are getting regulated, why we are seeing a fluorescence intensity increase at the region where they are interacting on the membrane. And to reconfirm, we analyze more than hundreds of vesicles having these morphologies across different batches. And what we always encounter is that whenever you increase the charged counterparts in your vesicles or in your condensates, you're always getting these sort of morphologies. And the other way around happens when you don't have charged particles, charged counterparts interacting with one another, you get budding morphologies. So since we saw this kind of a local fluorescence intensity increase, we were intrigued that does the lipid membrane properties also change in this region? So to reconfirm that, we do fluorescence recovery after photobleaching. So basically what you do is you bleach the region of your membrane and then you check for the recovery. So this is an indication of how fluid your system is. So this is basically a GUV membrane without any condensate on it, and you see a very beautiful recovery of fluorescence going on over here, and the diffusion coefficients are also pretty high. Now when you come to condensates, which are simply wetting on the membrane in case of budding, the T-halves or the recovery has slowed down a bit. Now, when you come to wrinkled membranes, where you have high affinity or a cluster of condensates on your membrane, you see that the recovery has slowed down quite a bit, and the diffusion coefficient has come down 10 times. So this does indicate that this high interaction of condensates on the membrane is causing the local lipid or the membrane properties to change. Now, we were interested to see what is actually going on, because until now we were just seeing an increase in fluorescence intensity at this local region. But what is the structure? What is actually happening over there? Is it lipid aggregation, or does it have something to do with the membrane itself? So we do confocal AD scan microscopy, 3D imaging, and we get very beautiful, intricate structures which show condensates sequestered or entrapped in between membrane folds. We also do transmission electron microscopy, which also shows similar kind of structures. So based on this, we call this process as membrane wrinkling. And based on the results, we propose a model in which we explain how even membranes can regulate the size of condensates simply based on electrostatic interactions. So what we say is basically, once you start your condensation, the nucleation happens on the membrane and membrane wrinkling starts. And once you go wait for more and more time, the number of wrinkles or the number of membrane folds increase, and the condensates which are getting entrapped in between are not able to fuse with one another, even though they are close by each other. Their sizes are getting regulated. So this was something very new, which has not been previously seen in literature. People have seen budding or wetting by condensates on membrane surfaces, but something like this was never done before, especially assembling condensates inside the vesicles and getting these kind of morphology. So this was pretty interesting. So coming to conclusion, what we basically found out from our study is that condensate size and surface charge are responsible for their lipid membrane interactions. Lipid bilayers are able to nucleate and stabilize small condensates by restricting their fusion. Strong binding induces curved, coacervate-rich membrane architectures in vesicles. So this mechanism can maybe offer insight into how condensates are localized inside cells or how their sizes are controlled within the cells. So if you're interested, you can also check out poster number 16 by my colleague Gitanjali Sahu. She's now looking into how charge screening or ion concentrations regulate electrostatics, and that in turn, how does it regulate the kind of interactions that we are seeing right now. So I would like to thank my supervisor, Dr. Harsha Bajaj, my institute, and the organizers of this event for giving me such a wonderful opportunity. So we are basically a membrane biology lab. We also look into aspects like membrane transport and membrane biophysics. If you're interested, you can check out these other works from our lab. So yeah, thank you, and I'll be happy to take questions.
So what's the ATP doing? Is only providing an electrostatic environment.
Sorry, sir?
The ATP?
Yeah, so basically ATP and polylysine is a complex coacervate system. So that is basically providing a negative charge. Yeah. So basically in cells also, it is the LLPS systems are formed majorly because of electrostatic interactions between charged biomolecules. So here also it is doing a similar job.
The sort of lowering of diffusion is coming because of the folds that are there in the membrane?
We hypothesize it like that because we see membrane folds in our 3D imaging, and we see condensates in high proximity with each other, but then they are restricted by a membrane fold, because of which they are not...
There's been a lot of theoretical work on studying the lowering of diffusion coefficient when you have folds.
It's not done theoretical work. All this...
No, I'm just telling you that there are lots of things you might like.
Yeah.
I guess one thing, like the system's very controllable, right? So you can change parameters quite easily. So have you done things like tuning temperature or so on to see how that impacts behavior?
No, we have not done anything with temperature.
It's all done at room temperature, is it? Or is it 37?
Coacervation actually occurs at ambient temperature. So I think if you change temperature, this condensation process might itself not occur. Yeah. So like that is also like if you give ambient buffer and temperature conditions, only condensation might occur. But no, we have not looked into how that might affect the kind of interactions we see.
Any more questions? Okay, so let's thank one more time.